Hello, chemistry family. We are going to start talking about the basics of any science class, and that, of course, is the scientific method. Something we cannot overlook and we cannot ignore. Throughout the year, when we start doing our experiments, we want to make sure that we can compare our results with something that is controlled, a comparison group, something that's going to allow us to rule out other things so that we can truly understand what changes are being made from what variables. But to do that, a basic process that you've already learned about before, most likely, is of course this scientific method. The idea that we have a few steps here that we need to follow. First of all, any natural thing in science starts with a question, and that comes from an observation. We want to recognize that there is some type of problem, and we often ask that naturally for you and I in the form of a question. Why does something happen the way it does, etc.? From there, you naturally think of a reason, an idea as to why that might be happening. And we know that as a hypothesis. Now, in science specifically, a hypothesis is a legitimate proposed solution, something that has some backup to it, maybe through experiences, maybe through data. All right, but the important thing is that your hypothesis has to be testable, meaning you have to be able to have an experiment to do that, to be able to find out if the hypothesis is correct or not. And that naturally leads then into the experimentation part of the scientific method, where you're trying to actually figure out and find if your hypothesis is correct or not, meaning did you find the answer to your original observation or question. Now, after the experiment, you do get results. But I think sometimes we think that after one experiment, you should get the results, your answer is done, and you're good to go. But in true actual science, that is not always the case. A lot of times when we set up experiments, we start to find weaknesses and flaws in our experiments. We have to start over. We have to tweak them to make them better and more controlled, which is something we'll talk about on the next slide. But regardless, after experiments, you'll get results. But you have to then start to think about how valuable are these results and how reasonable are these results, how accurate and precise are these results, how realistic are they, because you want to always pick apart your experiment to make sure the results you're getting are really focused on what you're looking for. So in that case, let's go to controlled experiment to really see what I mean by that. In order to have a proper experiment to actually get information that we really are looking for, all right, we want to have that controlled experiment, all right? And the control specifically are all the things that are the same throughout in the experiment. They're held constant. So in class, we may do a brownie lab. In that lab, we're going to keep the brownie recipe the same for everyone. Same pots, same pans, same amount of sugar, same butter, same degree oven, same amount of cooking or baking time. Everything we're going to try to control except for one thing in each batch. And that change for each batch of brownies is going to be the variable. So in one group, you might do everything the same as the regular brownie group, except for one thing. You might take out the sugar, or you might leave out the butter. But by doing that and only leaving out one thing at a time, whatever effects there are on the brownies, you will see, because everything has been the same or controlled except for the one change, which is the variable. Why is this important? Why is one variable important? It's so important because if you change two things, for example, if you were trying to see the effects of sugar in brownies and how fluffy they were, and you did the brownie recipe the regular way to see what regular brownies look like, and then you did a recipe where you took the sugar out, but you also left eggs out, and then the brownies are all flat and nasty, how can you honestly say whether it's the eggs or the sugar that made the brownies flat, or was it both? By only changing one thing at a time, we can zone in on that object or that variable and really see the effects of it. So that's something we'll have to keep in mind throughout the school year, that we want things as controlled as possible and that we have a comparison group, all right? We've talked maybe about Diet Coke and Mentos. If you haven't seen the videos or know the phenomenon, you put Mentos and Diet Coke and a huge geyser effect of soda and gas shoot out and spew out. If you wanted to test why Diet Coke and Mentos does this, all right, we would have to figure out a way to get variables, meaning we have a controlled experiment. So from a Mythbusters video back in the day, and we may have watched it in class, you may have seen it, they took seltzer water that just had carbon dioxide in it, and they used that for their control. And then they started adding different ingredients that are found in like Diet Coke, like aspartame and potassium benzoate. So by adding one ingredient at a time to this seltzer water and comparing the seltzer water with Mentos dropped in it to the seltzer water with one extra ingredient and a Mentos dropped in it, you created this controlled experiment where you could pinpoint and see what each individual ingredient did to the geyser effect of the Diet Coke and Mentos. All right? Either way, just so we keep clear and think about it the entire year, controlled experiments are very important. Throughout the year, we will talk about the scientific method. You will engage in that. But of course, this is a starting foundation for the course. So please make sure you do understand the parts of the scientific method as well as the parts of a controlled experiment, the difference between a control and a variable, and also the importance of why we need controlled experiments. Thanks a lot, chemistry family. Have a great day.